My name is Brian Keith, and I'm in the Department of Cancer Biology and also an Associate Director of the Abramson Cancer Center here at Penn. And today I'd like to talk a bit about how modern genomics and high-throughput DNA sequencing is changing our understanding of cancer. So to do that, I want to spend a few slides talking about cancer both as a clinical problem and as a biological idea. And then we'll get into a little bit about the specifics of high-throughput sequencing and how it's changing our view of what cancer is, and also informing new opportunities for treatment and therapy, both what the successes are and also what some of the challenges are. So let's just cut right to it. This is from a recent paper showing the actual cancer mortality as a function of time for the US population, and you can see that there was a peak in cancer deaths in 1991. Since that time, there's actually been a 22% decrease in the number of per capita deaths due to cancer. So that's a great story. I'll get into the specifics of that in a moment, but it tells us that this is although cancer is a constant problem and inherent part of our biology, that we can do a lot to actually battle this. If we break it down according to gender, this now shows data from 1930 up to 2011 showing mortality from different kinds of cancer. And you can see this big red line in the middle, and that's lung cancer. And there's some really interesting data hidden in this slide. One thing is we're starting at 1930, and at that point lung cancer is one of the least common forms of cancer, whereas stomach cancer is one of the most prevalent. Stomach cancer came flying down over the next few decades as improvements in nutrition and food quality came about. And interestingly, people really started to smoke cigarettes, which is the cause of most of the lung cancer deaths, back around World War I. And interestingly, the incidence of lung cancer associated with smoking didn't really show up until about 20 to 30 years later. And there's an important lesson in that. It takes about 20 to 30 years for most solid cancers to actually become clinically relevant. So, in fact, smoking really started to decline in the 1970s, right around here. But you see there's about a 20-year lag between the time at which smoking started to decrease and we see a concomitant decrease in the cause of lung cancer. So there's a lot of information in that because it tells us a lot about what it takes to become a cancer, how long it takes, and we'll get a little bit more into the sort of specifics of why that is in a moment. But as you can see from this decreasing line here, we're doing a much better job in terms of keeping people alive, partly because they're simply not smoking. A lot of this is prevention. Here we're looking at prostate cancer in this brown line here. A lot of that is due to screening, not entirely, but a lot of that is due to screening. Same thing with colorectal cancer. So for these three very prevalent forms of cancer, we're seeing great increase in terms of, keeping people alive, largely due not so much to intervention per se, but rather to screening and also to sort of lifestyle choices. And that's all very good, but you can see these other forms of cancer here. The rates of death to that, such as pancreatic cancer, leukemias, are pretty much flat. And we'll come to why that is in a moment. And just to be balanced here, here's the numbers for women. And you can see, for example, breast cancer deaths have been pretty flat until about 20 years ago and have really started to decline. In fact, now the 5-year survival rate for breast cancer is over 90%, which is really pretty remarkable. Interestingly, the number of deaths due to lung cancer has never gotten as high as it did in men, partly because women didn't start smoking until much later, and you can see that reflected in when lung cancer started to increase in women. And it's also just starting to turn down now due to that 20-year lag. So it's a changing problem in terms of what we're facing in the clinic. And we're seeing some successes, but what we really need to be able to do is not only avoid cancer and find it earlier, but actually be able to treat it and to treat it more effectively. And to do that, we need to understand what it is that we're fighting. So I want to spend a few minutes talking about cancer itself. We tend to think of cancer as one thing, and it's not. It's hundreds of different diseases with similar kinds of features. That is unregulated growth of tissue. And it's important to remember that it's not just the cancer cells, but in fact, tumors are very much like aberrant organs. They have transformed cancer cells in them, but they're also comprised of normal cells that give structure to these tumors and are actually, the way I think about it, they're almost hoodwinked by the cancer cells into helping the tumor grow. The stromal cells, the normal cells, sort of respond as though they think the tumor is a wound that needs to be healed, and in doing that can actually contribute to tumor growth. So there's an important set of conversations that go on between cancer cells and normal cells that lead to tumor growth. Interestingly, it's rarely the primary tumor that ends up causing death, but rather organ damage due to metastasis, that is the spread of cancer to other sites in the body that typically cause death. And that's due to obstruction of critical organs, obliteration of the bone marrow, for example, loss of immune responses, wasting due to the fact that cancer cells take up so much of our nutrients, and just physical breakage of blood vessels and the like. So it's a complex disease, and as I said, it takes, or set of diseases, it takes a long time to form. And in some ways that's shown nicely in this slide, which is just a series of micrographs of the cervical epithelium going through various stages towards becoming cervical cancer. So on the left is the normal tissue, and it shows the stratified squamous epithelium that is sort of like our skin in general. And as we can see, as these cells become more and more transformed and more unlike the differentiated cell type, they really become something that looks less differentiated, and there's a typically inverse relationship between differentiation and transformation. That is to say, the more sort of aberrant or malignant a cell is, the less differentiated it tends to look. But we can actually break this down into stages, and it sort of, again, it's a bit of a lesson that it's not one thing happening here, it's multiple events over time. And we can see that reflected in the changing and more aberrant phenotype of these cells, to the point where at this last stage, this cervical intraepithelial, within the epithelium, neoplasia, new growth, this sort of incipient tumor, it's really very different. It doesn't look anything like what we started with. But at this stage, this is still curable by surgery because it's still contained within the epithelium. And so when women have pap smears, really you're looking for these kinds of aberrant cells so that a surgeon can go in and remove that. The problem in cancer is when this happens. And here we're seeing these cells now crossing the barrier between the epithelium, where they should be, and the underlying stromal cells where they shouldn't be. And once these cells have become so aberrant that they can move across that barrier and gain access to blood and lymphatic vessels, they can now spread around the body and become metastatic. And when we talk about cancer, we're really talking about that process, invasion and metastasis. Benign tumors are ones that are contained and can be removed surgically. Malignant tumors are ones that are invasive and metastatic. And typically, by the time a person arrives in the clinic with symptoms related to cancer, usually the cancer is at a pretty late stage and has long since become invasive and metastatic. And once that's happened, although surgery is an important part of treatment, we can't cure people by surgery because the tumor has spread. So that's when we get into the need for other kinds of therapies. So what I'd like to do is talk for a few slides about how we got there. How did these cells become so aberrant and become so dangerous and capable of moving places they shouldn't be and surviving in areas that they should never survive? It really comes down to the fact that cancers are diseases of altered gene function. And the things that go wrong over those 20 years, we can think of in one way as accrual of mutations in specific genes. We'll talk a lot about that as we go through. Now, we've all heard about inherited predisposition to cancer, BRCA1 in breast cancer, and many other examples. And it's absolutely true that germline mutations, things we inherit, can in fact predispose us to cancer, but they don't determine it. Cancers always arise as a combination or accrual of mutations in somatic cells, that is, cells of our body. And it could be actually DNA changes or changes in the epigenetic structure of the cell, so that it really changes in gene function. And it's a combination of those changes within somatic cells that gives rise to cancer. So what are the sources of those changes? How do those mutations come about? And there are many. We talked about smoking already, and that's a classic example. There's 4,000 different chemicals in cigarette smoke, 60 of which are known carcinogens. So there are environmental factors, viruses that are associated with cancer, HPV, for example, cervical and head and neck cancer. Radiation, is another source of mutations that can lead to cancer. But just in being alive and having all the cells turn over in our body that do every single day, hundreds of billions of cells turn over just in our blood lineages every day. Every time a cell divides, there's a small but non-zero chance that there's going to be some DNA damage that occurs, a mistake in replication, a mistake in DNA repair. And when that happens, then you have a new mutation. And over time, as all those divisions occur, you're picking up more and more mutations. The vast majority of those are not a problem. But in rare cases, you get combinations of mutations in a given cell that together can act to overcome the normal regulatory pathways that keep cells doing their job and not turning into cancer cells. Okay. So this all leads to a kind of a model that cancers start with one renegade cell, as Bob Weinberg likes to call it. Again, the idea that mutations and changes in gene expression can happen in our cells all the time, but don't result in disease. It's just in that rare case where you get the right, or in this case, wrong combination mutations in a given type of cell that can lead to changes that ultimately overcome the normal controls on cell growth and division, cell survival, and motility. And those are really the things that change in cancer. And so it's those cancer-causing mutations that bypass that normal homeostasis that cause this to be a problem. Okay, so this is sort of the textbook model. And we're going to come back to this because I'll tell you right now, this is not quite complete. Okay, but we've really thought about cancer for many years now as kind of an evolutionary process, almost a Darwinian process. And if we imagine here's a single cell line of cells, maybe this is a single cell epithelium, perhaps the lining of your pancreas. And these cells are doing the normal renewal that they should and turning over the normal rate and differentiating. And this little white cell here happens to pick up a mutation that makes it divide too much. So it's dividing too much, but there are still all of these sort of homeostatic regulatory mechanisms in place that tell these aberrant daughter cells that shouldn't be there, you shouldn't be here. So these cells will undergo programmed cell death, or apoptosis. So you never see it. It never turns into a tumor because the cell is simply dividing and the progeny are dying. It's dividing, dying, dividing, dying. Not a problem. Until the cell picks up a second mutation at random that somehow increases its ability to either divide, and now it's turned orange, as we can see, or survive. And now you get an outgrowth of that. And the same thing occurs over time. There's a balance between cell proliferation and cell death. And over time, it'll pick up yet another mutation that turns it purple, and you get this clonal outgrowth. This has sort of been the model, and that it takes a long time for, and many mutations to accrue, for this to become a clinically relevant tumor. We can't even image tumors that are less than about 10 to the 6th to 10 to the 7th cells. And to go from one cell to that, there's an enormously long natural history. And that's the 20 to 30 years I was talking about. Okay. So this is one way to think about it. But as you can imagine, if this is true, then the cancers should look like all the same thing. They should all look like purple cells. As we'll see later, it's not that simple. It turns out to be more complicated. But the idea of picking up additional mutations at random over time and having that contribute to tumor formation is certainly correct. Okay, so which genes matter? And what are the kinds of mutations? And really, I'm summarizing about 40 years of research in one slide here. But much of the field has been focused on this, how to identify the genes that are really involved and really driving human cancer. And they tend to fall into two classes. And the first are things we call oncogenes. And these are just our normal genes in which some sort of gain-of-function mutation has occurred. Okay, so that might be an amplification of a gene, overexpression of a gene, or perhaps a mutation in the gene that leads a protein to be constitutively active. You might imagine a receptor on the outside of a cell that now can get activated in the absence of its ligand, or an enzyme that's on all the time when it should be regulated. That gain of function, if it contributes to tumor formation, we think of that, the gene encoding that, as an oncogene, so it's a gain of function. In contrast, the other side of the equation is a tumor suppressor, a gene which, by virtue of loss of function mutations, relieves a brake on tumor formation. And really, people tend to think of that analogy. An oncogene is like putting your foot on the accelerator pedal all the way to the floor, and a tumor suppressor is like losing a brake. This would be a gene that encodes a protein that might inhibit the cell cycle if something's wrong. or like P53, which is a protein many people have heard about that keeps sort of guard over the quality of the genome. If you lose P53, you might then replicate a cell that has a lot of DNA damage and increase the mutational load that can contribute to tumor formation. So it's really these two kinds of mutations, and there are many, many different genes that fall into both camps. And as I said, much of the work of the last 4 decades has been identifying which genes in our cells do that, which genes are involved in being oncogenes and which in tumor suppressor, and how do they work together. Now, many of these were identified in the 70s by virtue of being associated with viruses in other species, chickens and rodents. In fact, those viruses don't cause cancer in humans. We've used cell models and animal models and human tissues, and we've identified a lot of oncogenes and tumor suppressors, and probably most of the important ones. And so we sort of understand how they work. But what we've learned from that is that typically, and this is not to scare anybody, but typically what the proteins that are encoded by these oncogenes and tumor suppressors do, they're almost always involved in some aspect of interpreting or responding to signals. And what's shown here, and this is from a very famous review from Bob Weinberg and Doug Hanahan 15 years ago, and it shows everything in red is the name of a gene that's either an oncogene or a tumor suppressor. And if you can look at the cell here, you realize that many of those are on the outside of the cell. Those are receptors. They're seeing signals. Some of those other red proteins are in the middle of the cell, and they're involved in transducing that signal, passing the signal down through the proper channels. And many of these responses lead to changes in gene expression. You can kind of see all roads leading to changes in gene expression here. And in fact, some oncogenes like MYC or tumor suppressors like P53 are themselves transcription factors. They bind to DNA and activate genes. So in fact, really, cancer in some ways is a disease of signaling. Cells have to be told what to do, when to appropriately divide, differentiate, survive, and die. And those are exactly the kinds of things that are disrupted in cancer because the function of these genes has changed. So we know a lot already. We have a pretty good idea of what some of these genes do and how they work. So one of the big questions has been, all right, we know what we've seen. We're looking under the lamppost. Okay? We've been able to find things from clues from viruses and cell culture models, but we need to look more broadly. And basically, what are we missing? And this is really where it comes into the world of genomics. And so I'm going to spend a few slides talking about a really remarkable program called the Cancer Genome Atlas. And this is where genomics has really had a huge impact and continues to have a huge impact in cancer biology and how we think about it. And the idea was simply to take as many cancers as we could and to sequence their genomes and compare that to the normal genomes of the same patients. And it was called the Cancer Genome Atlas because biologists are hilarious, and they thought TCGA, which is, of course, the 4 bases of DNA. So anyway, that's biology humor right there. But this started in 2006, and it has been mostly funded through the ARRA. It's been a lot of money. It's required tens of billions of dollars to do that. And if you go to the website, which I recommend anybody who's interested to do, it's fascinating, it has all of the information. And so between 2006 and last year, they managed to, this is different sites around the country, using surgeons and biologists who could then convert surgical samples into DNA, do high throughput sequencing, then do the bioinformatics to interpret it. 11,000 different tumors, and where possible, matching DNA from the same patients was evaluated, and all of it has been made public. And it has allowed us to now look across a wide spectrum of cancers and many examples within a given kind of cancer. And that's really the important power of it, to try to find out what have we been missing, what's floating below the surface, what's not under the spotlight that could be important. Okay. And this is just from one of the pages in the TCGA website showing some of the different kinds of cancers that have been evaluated. Again, 33 different kinds of cancers have been evaluated. And this is a Herculean effort, but what it's allowed us to do is see, compare normal tissue, and everybody's normal DNA is a little bit different. So if you see differences between different tumors, do you imagine that those changes are important, or do they just reflect the background? by looking at both tumor and normal tissue, we can answer a lot of that question. And this just comes from a recent thing showing that the TCGA study of different human prostate cancers revealed that in fact there are 7 distinct subtypes. That never shows up clinically, but at a molecular level we can distinguish, we can subtype at least 7 different kinds of prostate cancer. They're different because the genes that are mutated in the tumors are different. And that dictates how those tumors grow. If we know what's different about them at the genetic level, it allows us to pick our targets, pick our treatments in a way that's appropriate for that subclass. And that's really the power of this. Right now, if we treated all breast cancers the same, it's not surprising that they're not all going to respond the same. We know there are many different kinds of breast cancer. And frankly, this is going to be true, I think, for every kind of cancer we can talk about, is that there are going to be subclasses of different kinds of the same, different kinds of kidney cancers, different kinds of liver cancers, and we need to know what those differences are. And one way to do that is just to do this deep sequencing and compare it in such a way that we can find what's different and then what's common, even at low frequency, between different tumors. Given how odd tumors are and how mutated they are, you might think it would be difficult to find the needle in the haystack. If you look at enough, you can find it. And that's really been the power. You have to look at hundreds and thousands of tumors to do this. So what have we learned? And this is from a review paper from Bert Vogelstein a couple of years ago, which very nicely captures a lot of the data from the TCGA. And what we're looking at here on the y-axis are non-synonymous mutations. So what that means is mutations in the protein coding regions of genes. So we're just looking really at 2% of the genome here, just the part that encodes proteins. And we're ignoring a lot of the rest of the genome, but this is among the most important. And what we see here is that depending upon what kind of cancer you're talking about, whether it's in adults or in pediatric patients, and whether it's in internal, different kinds of solid tumors associated with, in some cases, mutagens like lung cancers and melanomas. And if you look at lung cancers and melanomas, now of course most lung cancers, not all, are associated with smoking, exposure to a lot of mutagens. Melanoma comes from skin exposure to sunlight. And again, UV can cause a lot of DNA damage. Not surprisingly, if we look at the number of mutations in the DNA, in the protein coding regions, it's pretty high. We're up about 150 changes per different kind of cancer cell. For other kinds of cancers, the average is about 60 or so changes, single base pair changes. This is changing an amino acid in a coding region across the entire genome. So that's all 20,000 genes. So it's not a huge number, but it's a significant number. Interestingly, and again, perhaps not surprisingly, pediatric tumors show a very low level of DNA-based changes. Now, there could be changes in gene expression due to epigenetic changes, and we know, in fact, that's true. But these patients simply haven't been around long enough to pick up the number of mutations we see in adults just by dumb, random bad luck. So there's a range of these different mutations that we see in different cell types. And the question is, which ones matter? Right? We know about some of the genes that we've been studying for 40 years, but how do we think about the rest of the mutations? And analyses I won't have time to get into show that, frankly, almost all of these different changes that we see in these cancer cells have nothing to do with the cancer. They are so-called passenger mutations. Then, in fact, if you look in the normal tissue of the same patient, you're going to see the same kinds of mutations. The longer you hang around, if you sequence the DNA of my normal tissues, you'd find a lot of changes over time. That's just the normal biology. That doesn't, they don't typically lead to cancer. Normal cells pick up these same mutations all the time. They're either tolerated, or if it's a problem, all those homeostatic mechanisms are intact, and if there's a problem, that cell is taken out of the program through apoptosis, through programmed self-death. Okay? The problem is cancer cells have become typically resistant to normal apoptotic stimuli, and so they fail to die when they shouldn't, when they should die, and so they survive with this extra mutagenic load. So one of the questions is, can we use all this sequencing information to identify not just the passenger mutations, but the ones that really matter, the driver mutations, the things that are actually making the cancer cell a cancer cell? And can we use this information to find them, and then once we know what they are, be able to focus our treatments accordingly? So I want to give one example of the kind of data that come out of this genomics approach to cancer biology. And I could have picked really almost any cancer, but I've picked renal cell carcinoma. It's something that we study in our lab. And this is a paper that came out in Nature about 3 years ago now, and they did what they typically do. They took both tumor DNA and matching somatic germline DNA, normal DNA, from over 400 patients, and they compared everything. They looked at the protein coding region, the so-called exome, microRNA sequences, single nucleotide polymorphisms, DNA methylation patterns, RNA expression, everything they could think of to sort of see what's different from tumor to normal and how does that work across 400 different renal cell carcinomas. And the kind of data that come out of this, and again, this looks sort of overwhelming, but it's not. It's sort of gives us a sense of what's different in the tumor. And this is just one figure from the paper, and I'm using this to make the point that we get a lot of information out of this. We're looking at just all kinds of how many changes across the top. This middle portion here, what we're looking at are mutations in specific genes. Now, we knew enough about renal cell carcinoma before this paper was published that we could have predicted that some, like this gene VHL, is a known tumor suppressor in renal cell carcinoma, and sure enough, we see a lot of mutations in that gene. We discovered mutations in genes we didn't know about that pop up with some frequency in these different renal cell carcinomas, gave us a new insight into other pathways that might be important. This panel down here shows gain of copy number or loss of copy number. Again, amplification of a gene, which could be an oncogene in red, or loss of copy number, deletion of a gene, which could be a tumor suppressor in blue. And again, we see loss of VHL, loss of other tumor suppressors, and gain of things like MYC that we know are oncogenes. It all sort of makes sense. But now we're able to look and see within tumors, if you have this sort of mutation in a given gene, is there a statistically significant higher chance you're going to see a corresponding mutation in another pathway? And that would suggest something different about the biology. We can, again, subclassify renal cell carcinomas based solely on these kinds of genomic data, and that gives us a lot of power. Again, one example of this. This is a particular biosynthetic pathway, really important, called the PI3 kinase, AKT, mTOR pathway. And these boxes here just have the names of the different genes that are in the pathway. Not really important other than, here's a receptor on the outside of the cell and here's the responder. Now, if you look at the frequency of either oncogenic in red or tumor suppressor in blue mutations in the genes in this pathway, it's not very impressive. 2%, 3%, 7% here. You may not take that out as being important, but what's really powerful about this approach is if you look across all those tumors and add this up, it turns out that 30% of renal cell carcinomas have some sort of alteration in that pathway. And that's telling us that that pathway is really important for at least a third of renal cell carcinomas. We can target this pathway. There are multiple ways of doing it. So if we now had DNA from a particular patient and his or her tumor had these kinds of mutations in that tumor, that might suggest we should try to target this pathway. If the tumor did not have these mutations, it wouldn't make sense to do so. So it allows us, based on just on the genomic data, to sort of pick and choose our weapons accordingly. And again, the power of this is by looking at enough different tumors within a subset. Now, this takes an enormous amount of work. There are, as I said, different centers that do the data, the specimen collection, the processing, the sequencing, which is an enormous effort, and then the bioinformatics is tremendous. So if you actually look at the acknowledgments in these papers, so this is sort of the beginning, the bottom of the page where they start the acknowledgments. This looks like a lot of authors, but then it goes on the next page and you can see, literally there's 2 columns of this. So this is a massive undertaking. It's been going on, as I said, for about a decade, but it's been incredibly valuable and has really, I think, changed how we not only think about the complexity of the disease, but how we treat it. Moreover, and I won't go into too much of the detail on this, not only can we look within tumor types, we can now start comparing across tumor types and seeing that perhaps there are pathways or interactions between pathways that pop up in different kinds of cancers that might give us an idea of what acts as a driver in unexpected ways that is sort of been conserved in different kinds of tumors. And so this is all happening right now. This is all really exciting and giving us a new view in. So going back to this model, tumor heterogeneity, sorry, I should say the original sort of Darwinian model, and coming up with a sort of one big purple tumor at the end, we know this can't really be right. Because pathologists for 100 years have known that if you look at a real tumor, it doesn't look like one bag of tumor cells that are all the same. There's tremendous heterogeneity within a tumor. And this is just a section from breast cancer, and this is from Bob Weinberg's book on cancer, The Biology of Cancer. And each of these sort of regions that are circled in color are breast cancer cells, but they all look completely different. It's from the same tumor, but they're not the same. Something has changed. So how do we get there? And what does this mean in terms of our understanding of genomics? This is also from Weinberg's book, and it suggests how this might occur, where again, you might have an epithelium on the top that picks up an initiating mutation, causing that cell to divide too much. It propagates that mutation, and a second mutation is picked up. But over time, as the mutation rate increases, you're going to end up with clones of cells that have a bit of a shared history, but then things that are unique to that subclone. And this is a model of what might arise down here. And this is really more what tumors really look like. That yeah, they're all related cells, and they all have some natural history in common, but they're actually quite different in appearance, and some of the mutations that occur in one clone will not have occurred in another clone. Moreover, these different subpopulations of a tumor are talking to each other, and some are working collaboratively, and some may be inhibiting each other. And so we need to sort of think about how that affects the way we both think about tumors and how we monitor tumor growth using genomics and what it means for treatment. And this is sort of the same idea from another paper shown in a kind of a linear form over time, the idea that early on you pick up mutations, but here's a sort of the most recent common ancestor cell, if we want to think about this in an almost evolutionary way, that over time, as they pick up more mutations, you're going to have the emergence of clones that are, again, related, but different. They're not identical. And if this line here shows the point of diagnosis and maybe the beginning of treatment, we're going to have multiple subclones of cells. They may behave to respond to treatment in very different ways. We may eradicate some clones, and other clones could grow out. So that there might be a response, and then as often happens, way too often it happens, that tumors grow back. What comes back is not going to be what we started with. We're going to have selected for subsets of cells in this initial tumor that are different, that are probably resistant to whatever we treated with, and we need to know what those differences are. So, but with high throughput sequencing, we can imagine getting that information. You can biopsy here, do the treatment, biopsy here, and do the same sort of deep sequence analysis, and look at metastases. How are they different? How are they similar? And this is leading to the idea that if we do biopsies, which happens all the time, that in fact, where we biopsy within the tumor is going to give us a biased view. And in fact, so now what physicians are doing is multiple biopsies from tumors. And I think the future, we're not doing this as much as I'd like now, but in the future would be do multiple biopsies from a tumor and sequence everything. So we get a sense of how, variable is this tumor? How many different subclones are we looking at? And what are the differences between them? So in the last couple minutes, I just want to talk a little bit about the implications for treatment here. So for the last 40 years, we've been basically doing the same thing. And it's very effective for what it is. And I don't mean to make it sound otherwise, but we can cut things out, we can irradiate them, and we can poison them. And that has been basically cancer treatment. And it's been very effective for what it does, although the side effects, as we all know, are pretty dramatic, or can be. And the idea is that it's all based, other than surgery, the radiation and chemotherapy are all based on the idea of causing massive DNA damage to the point where the dividing cells can't survive. Now, normal cells can mount a response, fix the damage, and go on. Cancer cells have lost those controls and are in some ways actually more susceptible to this sort of DNA damaging agent. And this is just showing a picture of cells and nuclei and DNA in blue being shattered over time by one of these chemotherapeutic agents, doxorubicin, which causes DNA damage. You can see the shattered genome. When it gets to be a point where the genome is so shattered it can't move forward, that's called mitotic catastrophe. And that's really what we're trying to achieve with radiation chemotherapy. But as I said, we do know a lot about the sophistication of a cancer cell. We know differences about signaling and what these oncogenes and tumor suppressors are doing. So the idea is if we can use genomics to figure out what's different, and we have a drug that hits the driver mutation, we should be able to eradicate the cancer. If the cancer is dependent on that driver and we can block it, then we should have an effect. So I want to show one example of how that's both been wildly successful and also what the challenges are. So this pathway right here, which you probably can't read because it's too small, called the RAS pathway. If you're teaching your classes, you've probably covered the RAS pathway at some point. Very important signal transduction pathway. It's mutated in a large number of human cancers, and there's a protein here called RAF. It turns out that RAF, a particular mutation in RAF, is seen in 50% of human melanomas, okay? And we know it's a driver mutation. It's a gain-of-function mutation. BRAF is an oncogene. So there's something about BRAF that makes melanoma cells, when BRAF is mutated, turns melanocytes into melanomas. And 50% of human melanomas have that. So wouldn't it be nice if we had a drug that would target just the mutant form of BRAF? We do. In fact, it's an amazing thing. It's called vemurafenib, and it targets just the cancer-causing form of BRAF. And here what we're looking at is a comparison of treatment between a DNA damaging agent, which is the normal thing we use in the clinic, in this gray line, and vemurafenib in this black line. And what we're looking at here is time to tumor progression. And as this line drops, that means more and more patients have their tumors beginning to grow in the presence, even though they're being treated with a DNA damaging agent. The vemurafenib line here looks like a cure. And for a while, it looked really, really good. It looked like, yes, we can treat the driver mutation and it works. The cancer cells go away. The problem is that tumors are heterogeneous, right? And if you go over time, yeah, there's a real effect, but over time you get resistant disease coming out. And that's called acquired resistance. And I'm just going to show you an example of one patient because I think sometimes a picture really is worth 1,000 words. And there really is some important information here. So this is an example, this is from a publication 5 years ago, of a 38-year-old man who had malignant melanoma, and all these bumps on his skin are melanomas. Okay, this poor man was just riddled with the disease. And he didn't really turn gray, this is just a bad photograph, but his tumors had the BRAF mutation. So he was a good candidate for vemurafenib. He was treated with vemurafenib for 15 weeks. And you can see what looks like an absolute miraculous cure, right? And it proves that what I said is true. If we know what's wrong and we have a drug that can hit it, we can have a huge effect. But I already told you that these tumors are heterogeneous. They maintain treatment of vemurafenib. 8 weeks later, he's got tumors again. But what's really, really important to notice here is if you look at the pattern of the tumors, it's exactly the same. Right? The tumors that were here went away, they came back in the same spot. What that means is that even though 99.9%, I'm making that number up, but of the cells that were in these tumors were sensitive to vemurafenib and went away, that because of the heterogeneity of the tumor, there's always some number of cells in that tumor that are already resistant, and we simply selected for them, and now they're growing out. Okay, and unfortunately, this gentleman can, succumb to his disease. But what this means is that, yeah, we know what to do. If we can find the driver mutation, if we're lucky enough to have a drug, we can have the effect, but we have to anticipate this acquired resistance. And if we can go back in and now biopsy some of these tumors, use the high throughput genomics, figure out what's different about them, how that has changed, and then hopefully have a drug that can treat that. And really, I think this is in some ways the future of cancer. Cancer's never going to go away. It's part of our biology. But what we can do is try to stay one step ahead of it and turn what used to be a death sentence into a low-level chronic disease in a perfect world where if it comes back, we can sequence it, figure out what's wrong, and then be able to treat it. And my last 2 slides, just to make the point that this has been seen with every single targeted therapy, and there are many good, effective targeted therapies out there. We always see some aspect of this acquired resistance due to the inherent tumor heterogeneity. It means, among other things, we have to have cleverer ways of treating with combinations of therapies, chemotherapy, radiation, surgery, targeted therapies, immune therapies, different therapies, how we're going to figure out using HIV as a paradigm. You use a drug cocktail with HIV because 1 or 2 drugs isn't enough. To overcome the resistance there, you need to use multiple drugs. It's the same thing in theory, sort of in concept, I think, with cancer. And what's really exciting is that there are new therapies on the horizon. They're actually being used right now that are based on some of the fundamental differences between cancer cells and normal cells having to do with their different metabolic wiring, and also the fact that for a cancer to form, it has to find a way around our own immune systems. Our immune systems are very good at finding cancers because they look different, they look foreign, and getting rid of them. For a cancer to form, it has to find a way around the immune system. And so there are multiple different ways that people are now taking a patient's own immune system and either rewiring it or re-energizing it to go and fight the tumor in a better way. And that's turning out to be extraordinarily successful, even with things like melanoma. But at the end of the day, we're going to need to know what's growing out, how is it different, and this is where genomics is having such an impact. The ability to, within a couple of days, sequence an entire genome and get that information is going to save people's lives. We don't have it yet as something that is sort of standard treatment, but I think that's something that is achievable. And this is just my last slide. Getting back to the idea of cancer immunology and immunotherapy, this is an extraordinarily exciting time for this, and a lot of it's going on here at Penn. And maybe they'll have me back and talk about that some other time. So that's what I want to talk about. There's my email address. If you have any questions or if I need any clarification on what I've told you, don't hesitate to get in touch.